Awesome. Yeah, thank you to the organizers for having such a great group of talks on these 2 days here. Yeah, my name is Teague Tomesh. I work at Infleqtion. Today I'll be talking about our Welcome Leap Q for Bio project. This is titled Quantum Biomarker Algorithms for Multimodal Cancer Data. This is a collaborative project between folks at Infleqtion, the University of Chicago, as well as MIT. And our project is focused on developing quantum classical algorithms to do biomarker discovery in cancer data. So I'm not a biologist. I don't know if there's any biologists in here. So first things first, what is a biomarker? So a biomarker is a measurable feature or set of features that indicate clinically relevant states or outcomes. So it can be important for a clinician or an oncologist to understand if there are particular mutations in a patient's DNA that make them more prevalent to certain types of cancers in different tissues, or the holy grail here is if you could predict what treatment would be best suited to a particular patient given their DNA, their gene expression, what the tumor looks like, et cetera, et cetera. So to give a little more details about what our project is focusing on here, so we're going to focus on 3 different modalities of data. So these are gene expression data, the mRNA level, mutations in the DNA, as well as images of tumors or histology or pathomics. Current approaches to processing these data sets are limited sort of in 2 main ways. So on the one hand, they may throw very complex, large models at these data sets. This can lead to issues like limited interpretability and deployment in a clinical setting. If you can't understand why a model is making a decision, oftentimes clinicians will not be so happy to take that as gospel. On the other hand, we could apply very simplistic models, but these models tend to suffer in terms of accuracy when you're trying to perform some learning task on these data sets. And so we can outline sort of 2 main challenges here. One, these data sets, these multimodal cancer data sets, I think could be characterized broadly as being in this sample scarce regime. So these data sets may have many thousands of features, but the total number of patients that you're able to collect all of these different modes of data on tends to be quite small. This is just due to cost and time of collecting this data. And on the other hand, we would love to have a model or methodology of a way to explicitly incorporate higher order correlations that exist in this data, both within a single data mode and across these data modes as well. This is very computationally challenging, and this is an area where we are interested in developing applications for quantum computers to directly address this challenge. And in fact, we'll be focusing explicitly on this task of feature selection on these multimodal cancer data sets. So our project started out actually looking at sort of all 3 of these steps. If you can imagine this data processing pipeline for this multimodal cancer data set, there's publicly available repositories. We're also working with an oncologist at U Chicago, and they have the ability to go out and collect new data as needed. Once you have this data collected, oftentimes it needs some cleaning, some dimensionality reduction. So we can characterize that here as this feature selection step. And then once you've selected these features, this could pass down to some downstream model that's going to perform a learning task. I'll show you some examples in this talk where we're performing classification like cancer tissue of origin, and we're also taking steps towards predicting treatment, patient treatment response. At the beginning of our project, we sort of looked at applying quantum algorithms to both of these steps here, both the dimensionality reduction and performing the learning task. Throughout the course of over the last year, we sort of shied away from the learning aspects here. This is more due to time and limited resources than anything else. I think there are still some interesting questions to be studied there, but the majority of our project now is focused on developing hybrid quantum classical feature selection algorithms. So to give you a quick overview, first, I want to talk briefly about how we classically pre-process this data. And then I'll give you an overview of the algorithm that we've developed. I'll show you some simulation results that we've collected on Perlmutter here. And then I want to finish by addressing sort of our current and ongoing work, addressing the bottleneck in these variational algorithms that we're going to use. So to start, to give you a better picture of the data that we're working with, let's start with the DNA mutations. That sort of starts at the bottom of this biological pathway, right? All three of these modalities are biologically related to one another. The DNA mutation data is great. It's very well behaved. It's like the raw data comes as these binary features. Is there a mutation in this gene or not? The mRNA data is a little bit trickier to work with. So the raw data comes as continuous expression levels. For a specific gene, you can measure the relative level of expression of this gene. Originally, we wanted to discretize and sort of compactify this data for the purposes of loading it on a quantum computer. Since we shied away from looking at QML algorithms to do the learning, we found that it's actually still useful to perform this discretization. You'll see that we're going to do feature selection based on mutual information, and it's just very much more efficient to compute mutual information on these few discrete values than trying to integrate over these continuous values. So what we did, this is an example here for a particular mRNA feature showing distribution across patients. And we're just going to perform a quantile discretization here. So what we're left with is categorical features 0 through 4, denoting the relative expression of this gene. For the pathomics images, these are even a little bit trickier. So the raw data here for each patient is on the level of these gigapixel images. And to make matters worse, there's oftentimes a variable number of these features per patient, which can make feature selection very difficult. For this, actually, the team at UChicago has developed a very robust deep learning, classical deep learning framework. It's called SlideFlow. You Google SlideFlow, you'll find their GitHub page there. This is a very great deep learning pipeline for us. We can feed in all of these gigapixel images, and what comes out at the other end is a vector of a controllable length. It still has continuous values, and so we developed an add-on pipeline to this step where we discretize these values again using a quartile discretization. And what I'm showing here on the bottom right is just an example of performing this UMAP cluster visualization using the continuous features. And after we perform our discretization, we still see the same clusters in the data. So at the end of the day, what we're left with are binary DNA mutational data, categorical mRNA expression data, and categorical pathomic image features.
Can you tell roughly how big or what is the cardinality of those categories?
It varies based on modality. For the data sets that I'll be showing results for, there's on the order of 23,000 features in the full data set. And so the way we're going to frame a feature selection on these data sets is we're going to use a polynomial constrained binary optimization. This is a nice framework for us that allows us to explicitly capture higher order correlations in the data set. So we can choose to include first, second, third, fourth order terms, et cetera. In the work here, we'll only go up to third order. It's a constrained optimization problem. We are telling the problem that we want to select n features. It's binary. So this is an interesting point here that when we're mapping this problem onto the quantum computer, we don't need to worry about loading the explicit data values. We're only going to load, those will show up as sort of rotation angles in the circuit. The qubits themselves, the state of the qubit is only going to represent whether or not a particular feature is selected here. So there's a linear scaling with the number of qubits and the number of features that we want to select on. And then we're going to perform this optimization. We're going to try and minimize this objective function here. And so we have a freedom to choose how we want to define these coefficients that get applied to the different terms in our problem. And we're going to choose to define these coefficients using mutual information in the data set. Yeah.
So it means that you are assuming 23,000 qubits now.
So I have a slide here where we address this issue, right? We don't have 23,000 good qubits available for us to use. So how can we break this problem up into a constant size that a constant size quantum computer could solve?
I think confused because earlier you untie some of your features to do categorical features.
Yes.
Now telling me that the features are binary, but either I have or I don't. So what is that category do for you now?
So the xi variables here are binary, and that's going to represent our qubit 0, 1 state. Is the feature I selected or not? So the features, this f sub I here, those are still our categorical features, and that information will get encoded into the problem in these coefficients here. So to give you a concrete example, we might apply our first order features. We might have them weighted by the mutual information between this feature column and the label column in our supervised learning problem.
So is it like one hot encoding of 23,000 qubits?
One hot encoding. No, so the valid solutions will live in this subspace of all Hamming weight n bit strings. Or I guess maybe your question, we need one qubit to represent one feature,
Yeah, so
whether or not it's selected.
you need to focus on one hot encoding.
Sure, if you want to view it that way, yeah.
Okay.
But we still have this problem of I don't have 23,000 features to represent my data set. Absolutely. That's a great question. For the simulation results that I'll show, we're going to encode that in the cost function itself and turn it into an unconstrained optimization problem. We are also testing like a constrained mixer where we only evolve in this Hamming weight n subspace. Both are viable approaches.
Or again, is fi a feature or is one of the categories of a given feature
This fi is a feature column across our whole data set. So it's a single vector. Yeah. And then I'm going to compute a mutual information between that feature column, that feature over all patients, and the labels y over all patients. So maybe intuitively, so I'm showing here on the right, these are some other examples of mutual information based feature selection energy functions. These are nice intuitive ways to understand. So we call this MRMR for minimum redundancy, maximum relevance. So intuitively, you want to pick features which maximize their correlation with the labels, but minimize the redundancy of the features in the feature set, right? And intuitively, that's what you would want a good feature selection algorithm to do. We can extend this with this full QUBO definition here and this 3-body QUBO definition here, where we're going to include sort of higher order interaction informations between these features. There's a lot to explore and cover in just defining these problems as well. Awesome. Are there any more questions at this point? Sweet. Yeah, so these are challenging problems to find the ground states of these energy functions, and we're going to apply the recursive quantum approximate optimization algorithm to try and find approximate solutions to these problems. So we don't have 23,000 qubits to load our whole data set onto the quantum computer. So what we'll do is we'll actually divide up the data set and solve many smaller subproblems. The particulars of this approach aren't too important. So we refer to this as a PCBO tournament. First, we're going to classically pre-compute these large matrices that hold all the correlations between our features that define our problem. And then we're going to divide the full list of features into subproblems. We're going to solve this PCBO on each of those subproblems to select, usually within the feature, we'll select half of them to continue in the next round of the tournament. So we have these features face off against one another. And at the end, what we're left with is a single feature set of size no greater than our quantum computer. And then we'll solve that final PCBO to select the desired number of features.
So your features came from 3 categories of data.
Right.
So how did you divide this 23,000 features onto subsets?
That's a great question. For the simulation results I'll show you here, we were just doing random subproblem generation, which I think surprisingly we found still yielded okay performance. there's also a lot of design choices that can be made in that respect. So we've also tried clustering based on mutual information-based distance metrics. But this is an active area of continued research. What's a good principled way of generating these subproblems? Because we are giving up something where if two very important features that are correlated with one another end up in different subproblems, we have no way of capturing that. Yeah, so you're exactly right. In the interest of time, I think I'll.
You mentioned at some point that you were doing dimension correction.
Yes.
Do you use it here or?
So we've used dimensionality reduction to generate this input data set here. So now our features, the raw features, are no longer these continuous gigapixel images. They're columns of discrete categorical variables. So for the mRNA features, it was just a quantile discretization. For the pathomics features, there's a classical deep learning pipeline in there to extract a feature vector from the actual images. Cool. So here's some simulation results collected here on Perlmutter. So this is on the right-hand side. I'm showing feature selection on a pan-squamous data set. So the task here is predict and classify each patient. They may have one of three cancers: cervical, lung, or head and neck cancer. There are on the order of 2,000 DNA features here, 18,000 mRNA features, and 2,000 of those pathomics features, with only 660 patients. The other thing that I'll mention is that this raw data set is horribly biased in that actually the perfect feature to perfectly classify this data set is just what hospital was the data collected at. So to address this bias, we formed 3 different site stratified train test splits. And so that's what's giving us our error bars here. The algorithms are run 3 independent times on these 3 different data sets. We're reporting the average accuracy of a logistic regression classifier on the data.
Just to clarify, you're saying one hospital is better at predicting these cancers?
No, so for example, all of the patients with cervical cancer came from hospital A. All patients with lung cancer came from hospital B. So if you knew that feature, you could perfectly classify the data. Yeah.
So government software.
Yeah, exactly. Exactly. No, but it points to a real problem when working with these data sets. The way it's collected and how it's collected. It also shows up with the pathomics images. Some of these deep learning pipelines will tune into just the amount of dye that's used to dye the cells themselves, and that can vary across hospitals as well. Yeah. I learned a lot of interesting sort of cancer biology through this project. Yeah, happy to talk more about that. Yeah, exactly.
How many people did you select?
Great question. So what I'm showing here on the right, from those 23,000 features, we are selecting on this first column here, 6 features, and then here in the middle column is 10 features, and then 14 features. On the top is the weighted F1 score of the logistic regression classifier. On the bottom is the balanced accuracy. And I'll point out here that this horizontal line, this is the same logistic regression classifier trained on all of the 23,000 features in the data set. And so what's interesting is this red point here. So this is the full QUBO formulation of our PCBO. And actually, we're able to select a feature set of size 14 that is sort of actually significantly improving the performance of this classifier, which actually a priori we would not have expected that you could improve the performance if this model is given all the data. Why wouldn't it just overfit and perfectly classify? So this was reassuring to us that this approach sort of is a nice way to frame feature selection and maybe has some legs to it. So I'll quickly mention that we can also inspect these features. I don't expect you to understand what they mean. The thing that I learned is that actually this TP53 mutation is a very common and well-known biomarker for these cancers that we're classifying in this data set. Cool. the next thing that we wanted to look at was we want to know and understand how is solving our PCBO problem, finding low energy solutions to this problem correlated with the accuracy of the feature sets that are selected. So on the right-hand side, I'll start here. This is showing a scatter plot. This is actually for a different data set. This came from this I-SPY trial, which is actually getting at treatment response for breast cancer patients. And what we did here was we used 3 different formulations of the PCBO. The 2 on the left here only contain first and second order terms. And on the right, we're containing first, second, and third order terms. And what we found was that including these higher order, these third order terms, we can get a tighter correlation between the energy of solving our problem and the feature set accuracy that's produced.
With this correlation of 0.4 survive if you take out this 1 point on the top, right?
Perhaps. So this top point on the right is the best feature set out of this subproblem that we've generated. So I think you would want this feature set to be the one that has the lowest energy as well. So I think, yeah, absolutely. I think if you took that feature set out, all of the other feature sets are sort of equally bad. So you would want to identify this one, I guess is the point. But how can you actually find that one very good feature set? You need to solve your problem well. So on the left-hand side here, what I'm showing is 3 different classical solvers that you can use to solve these PCBOs. And the thing that I'll point out quickly is that, so this is showing on the y-axis minus the energy. So higher is a better solution to our problem. And solving the problem better correlates well with the feature set accuracy that is actually produced. So all this is to say is that this is motivating us to develop better solvers for these PCBO problems. And this is exactly where we would like to apply hybrid quantum algorithms. And so that's what we've done. We spent a lot of time sort of collecting simulation results on Perlmutter. And what we found is that indeed, RQAOA can be competitive with classical solvers at modest depth values of P. Modest here just mean that it's less than the size of the problem that we're able to study. So on the left-hand side, these are showing median approximation values of the RQAOA. Those are the blue points, and then we fit a linear trend to that curve. And then the horizontal points are 2 of these classical solvers. So QBsolve from D-Wave and Anil Pubo is an open source implementation of simulated annealing. And on the right-hand side, these are showing box plots of the same data, which is to say that there are some settings of the parameters which do solve these problems perfectly. The challenge is finding them. And so that's exactly what our current research is focused on. So we've seen that good parameters do exist, but it can be computationally demanding to find those parameters. And so I don't need to explain this variational loop. We saw it a lot yesterday, but tracking your way through this very non-convex landscape can be very challenging. I'll point out that one interesting new development sort of in this field of QAOA research is this idea of parameter transfer. So it has been shown and proven for at least QAOA on max cut for 3 regular graphs that you can solve your problem well at a small problem size, freeze those parameters, and take them to initialize a new QAOA circuit at a larger problem size and still retain or extract good solutions at that larger problem size. And so that's exactly what we're aiming to do now. We're asking the question, can we offload this expensive variational optimization, ostensibly going to be done on your quantum hardware? Can we offload that to powerful classical simulations at small problems, maybe on the edge of what you could feasibly simulate, take those parameters where we found a good local minima and use them to initialize a larger problem that we need to run on the quantum hardware beyond the regime of what we can classically simulate. So this is what we're studying now. I'm just showing here an example that I was able to collect for, this is depth 1 QAOA, so I can easily visualize it, but this is showing the loss landscape for an 8-qubit problem, and then the same loss landscape, or it looks similar for this larger 10 qubit problem. So this has been theoretically proven for max cut on 3 regular graphs. It becomes very difficult to sort of make these broad, provable claims for other problems. And so I think the best tool that we have here is just go out and empirically test whether or not we can find good solutions with parameter transfer. So with that, I'll leave you with this conclusion and summary slide. This work is still ongoing. We have an overview in Nature Cancer, and there's a new preprint. It's not on the archive yet, but it is my duty to get that up on the archive within the next month or so. So with that, thank you.
And just for one question, I think we'll be late. Right.
So the model that we got, how do you do that on D-Wave?
Great question. The embedding of that onto the D-Wave device, we're actually trying to get access to D-Wave right now so we can test that out. That's the question that I want to answer. They do have a lot of qubits, so they can afford some overhead, but at what cost and performance? I think this is an interesting question that I'm hoping to answer within the next 3 weeks or so. Yeah, in the circuit model, it's very natural to include these extra higher order correlations. You just increase the depth of your C0 ladder in the QAOA circuits.
Thank you.
Sweet.